So I'm going to talk about the scientific examination of Eucharistic miracles, particularly the phenomenon of bleeding host. So let me start this out by saying I'm a faithful Catholic. I've been a Eucharistic minister for over 20 years. How I got into the subject is I've done quite a bit of study on the Shroud of Turin. Number one question I would always get, well, what about the blood type on the shroud? It seems to be the same as what's been reported for all these Eucharistic miracles. Could there be some connection? So last November, I published a couple of articles. The first was on the shared blood type, so I'm going to talk about that first. The second was just kind of general properties of Eucharistic miracles. So here's a pie chart that shows a typical blood distribution among the population. There's going to be some variation depending on geographical region, but by and large, most people are type O. A close second place is type A blood. A pretty third distant is type B. The rarest of all is type AB, typically about 3 to 5%, again, some variation depending on the particular geographical location. Now, certain relics associated with Jesus, such as the Shroud of Turin, top left. In the middle there, the Siderium of Oviedo, that's a proposed face cloth. And then on the right, the tunic of Argentuli. This is proposed to be the robe that Christ wore on the way to Calvary. All of these type is AB, as well as all the Eucharistic miracles that have been tested to date also test as AB. So it's been said, well, there's no way this could happen by chance. It's a statistical improbability, greater than 3.2 million to 1. Some even put the odds in the billion to 1 range. A recent article in Catholic Answers magazine said, well, this is mathematical proof of the real presence, proof that God is real, proof that the Lord has AB blood. If you look at the structures of what the A and B molecules are, they're made of carbohydrates, or in other words, sugars. And if you look at this middle section here, you can... Oh, I'm sorry. If you look at this, thank you. If you look at this middle section here, you can see that that's identical in the A and the B. What makes it different is this carbohydrate that's on this side. So that's really the only difference between A and B. Now, what's often overlooked in these kind of discussions about shared blood type among relics is that many bacteria, many common bacteria, express identical A and B antigens. They're expressed on human red blood cells. The same identical molecules are also expressed on many common bacteria. Many of these relics and the miracles, they've been shown to have significant bacterial and fungal contamination. I think this is something that you have to take into account when you're trying to establish a commonality between all of these objects. The tests that have been done for this are immunological tests, using serological methods or antibodies. Antibodies cannot distinguish between bacterial AB and human AB because, again, the molecules are exactly identical. There are ways that you can sort this out with DNA methods, bacterial AB versus human AB, but that has not been done to date. I want to point out that shared AB, that is not scientifically sufficient to demonstrate the claims of a single origin among these various objects. To do that, you would need markers that are unique among individuals. So some that might do this, you could use nuclear mitochondrial DNA, just kind of a standard DNA analysis and profiling, forensic cases, things like that. You could also use the HLA transplantation antigens. Now what those are, if a person goes in to get a organ transplant as a donor recipient, they're typed to see if they match. The HLA has kind of an advantage over the other two in that you could use these at both the DNA as well as the protein level. So if you're interested in this, here's the reference for that. This is the paper that came out. Sorry, thank you. This is the paper that came out last November. The top one and the bottom one is one that I wrote before that just on AB and the shroud. So you can Go back to this slide for the reference, or just put in my last name in a search engine, and it should pop up. So now I'll just go into kind of the general characteristics of Eucharistic miracles. So as I mentioned, I didn't really know a lot about this going into it. My understanding at first was, well, the bleeding of the host typically happened when the priest said the prayer of consecration. But as I would read about several cases, a more typical scenario is like this. Someone finds a host in the back of the church in a dusty or a dirty area, shows it to the priest. It's too dirty for the priest to consume, so the church protocol there is you put it into water and dissolve it prior to disposal. These reddish spots did not start to show up until after being in water for some 7 to 10 days. So we were curious as to what would happen if we took non-consecrated wafers. I want to stress that all the experiments we did were using non-consecrated wafers. What would happen if we took those and put them under similar conditions as has been shown for these Eucharistic miracles? So here's a pretty famous miracle. This one's shown on the left. This is from Poland and Circulka. And this is what we found in approximately 15% of the cultures that we did, non-consecrated wafers cultured in water. This is what we got. So you can see there's some similarity in the appearance. If you look at the genuine miracle over here, you can see some dark spots. We also have similar dark spots in ours. Now in our case, we were able to identify these as fungal spores. One property that's been described for a lot of the miracles is that while it's in water, the water does not become tainted. Like that's sort of a divine property. It's kind of an odd property for blood to have, especially fresh blood, because you would think that that would mix very well with water. Now, in our case, the water did not become tainted either. In fact, if we took genuine blood, we put it on a non-consecrated wafer and let it dry really thoroughly, put that into water, it would very quickly dissolve. Now, this is shown for 72 hours, but it happens much faster than that. One thing that's been implicated historically in a lot of these miracles is a particular bacteria called Serratia marcins, which produces a red pigment very similar to the color of blood. Now, this is just one of a number of bacteria that can produce reddish pigments. There's also a number of fungi that can produce reddish pigments as well. But this is the one, if you look through the literature, this is one that's kind of been implicated the most. So we got some of this bacteria. This is a streak plate that we did. And then we intentionally inoculated non-consecrated wafers. So here's the result from that. Now, if you look at the appearance of these, and you compare that to a lot of the images that are on the internet of the miracles, there's definitely some similarities in appearance. We looked at the pigment under ultraviolet light and found that it did not fluoresce. It does have this in common with blood. So blood, genuine blood does not fluoresce under ultraviolet. Neither did the pigment. Now, if you go back to those non-consecrated wafers that you put in water, not inoculated with the bacteria, but the ones I was talking about just a few minutes ago, we found in that case, those reddish spots did fluoresce under UV, a bright orange. Again, in contrast to what you find with regular blood, which does not. Now, here's just a close-up of some of that fluorescence. Here's a higher magnification. So ultraviolet could perhaps be a useful discriminator in some of these cases kind of early in the game when they're first starting to be looked at. There's also been some DNA studies on some of the miracles. So for example, in Buenos Aires, it was reported there's a very low concentration of human DNA present. This is just consistent with normal handling or contamination. But they also said that the sample contained a good amount of DNA from a non-human origin, DNA that did not react with human-specific probes. So what their conclusion was, this DNA did not react with human probes. It defied detection because it was of a divine nature. Also in Tixla, Mexico, presence of genetic material was detected. Again, no known human coding sequences identified. It did not react with probes for human DNA, they said that the genetic material escapes genetic probes. Now, the faith teaches us, the catechism teaches us that Jesus is both fully human and fully divine, so you would think that he would have some human DNA that was present. In our case, with the non-consecrated wafers, we wondered, well, what type of non-human DNA might be present that could account for these kind of results? Well, the wafers are made of wheat, so using just standard PCR, DNA amplification, and sequencing techniques, we found that wheat DNA was present. We also found that DNA from numerous bacteria was present. And the strongest signal that we got in our case was fungal DNA, and particularly one fungus called Epicoccus nigrum, which produces a red pigment that fluoresces orange under UV. So in our case, we think this is what is responsible for the reddish spot. So here are 3 different sources of non-human DNA that would not react with human-specific primers, but in the studies, this kind of thing was never considered. It was just automatically jumped to, well, it has to be divine DNA that's present. So the purpose of the second article is not to say that Eucharistic miracles cannot happen. It's just to stress that the scientific rigor of these needs to be raised. Particularly, we suggest that a uniform standard scientific protocol be developed and suggest certain steps that can be followed uniformly. Also, the emphasis is made to make these scientific results more transparent. Instead of science by press release and putting on social media and things like this, have them go a more normal scientific route. Published in peer-reviewed scientific journals. Have that really be the end game in these types of studies. Now, there's more data in the paper, but if you're interested in that particular reference, that's shown here, so you can go back to this slide, or again, just put my last name in a search engine, it should pop up. Now, I've been asked to say a few things here at the end about the Shroud of Turin because there's some overlaps between the two, so I'm going to kind of finish up with this. The Shroud of Turin, some people believe that it's the burial cloth of Christ. Others think it might be some type of medieval forgery, just a hoax. The shroud has really only been investigated to any major extent twice. So one was in 1988 when they did the carbon dating. The most extensive investigation, particularly for the bloodstains, was done back in 1978, about 40 years or so ago. There was a so-called STIRP team, this stands for Shroud of Turin Research Project Team. It was about 40 scientists of various disciplines that were allowed to look at the shroud for about 5 days straight. There were also some additional scientists present as well. Now, what you'll usually hear about the bloodstains on the shroud is this. It's been investigated by scientists, and scientists have determined, they've shown that the blood is human, the blood is male, and the blood type is AB, although none of these 3 have ever been published in a peer-reviewed journal. I've already talked about the AB, so I'm just going to go to the other 2. To determine, for sex determination, you would go the DNA route. To determine the species, if it's human, you could go DNA or an immunological route. I'm going to start with the sex determination first. Back in 1978, when these samples were collected, this is what the state of DNA science looked like. You can see that nobody is wearing gloves. Here's one scientist. He's right over a bloodstain collecting a sample using both hands and also the mouth to hold a third instrument. 1978, it wasn't realized that you should take precautions with gloves and masks and things like this. As far as reports of the DNA from the bloodstains of the shroud, there's been 2 that came out in the mid-90s. Probably the most famous is from this book. It's called The DNA of God, written by Dr. Garza Valdez. He looked for portions of 3 genes. He looked for the X and Y for the sex determination, and then also the beta globin gene, which is a subunit of hemoglobin. He got a positive signal for all 3. So he concluded that the DNA was from a human male, that the bloodstains, the DNA from bloodstains, is from a human male. He said that he knew that the DNA was from the bloodstains because he got a positive signal for the beta globin gene, hemoglobin. Unfortunately, what he overlooked is that skin cells also contain the beta globin gene as well. So there's really no evidence at all that this signal truly was from bloodstains. Simple contamination could very easily explain the results. There was another study that came out in the mid-90s, this is in a Shroud magazine, where they looked at DNA from a bloodstain. There they found both male as well as female DNA that was present. When they looked at unique markers, polymorphic markers, they could account for the profile of at least 6 different individuals. So these results were considered scientifically null and void. I wrote about this a couple years ago. Here's the reference for that. If you want to go back to the slide, or again, just hit a search engine. And I'll finish up with this part here about the human designation. So one thing that's always a concern if you're working with DNA in the blood, well, is the DNA signal really coming from the blood, or could it be coming from contaminating skin cells? One person in a single day sheds about 10,000 or so skin cells, some of which contain DNA which can be transferred as contaminating or touch DNA. One way you could go about this is to look at immune receptor genes. Now, these are present in blood cells, but also skin cells. What makes it unique is in one type of blood cell, lymphocytes in particular, here the DNA actually undergoes rearrangement. The DNA is cut, spliced, and pasted back together as part of that cell's normal maturation and development. They're the only cell types in the body that do that. So you can have a probe against the rearranged form of the gene, and you know that the signal, what you're looking at here, is truly from skin cells. It would not matter how many hundreds or thousands of people had handled that particular relic. And this would also work in the case of Eucharistic miracles as well. And you would then take that DNA signal that you got, sequence this, and you could get some species information from it. But to date, this has not been done. What has been done are some studies using immunological methods. These were reported back in the early 80s. So these are studies using antibodies, serological methods. They took anti-human antibodies, and antibodies themselves are very, very small, so you can't see them with the naked eye. So typically what you do is you add a kind of fluorescent tag to them, or chemical tag, and then you look under the microscope, and wherever this lights up, you know that the antibody has found its particular target. So what they did, they took unstained fibers from the shroud, fibers without blood. They took blood-stained fibers, and they reacted them with this antibody that has a fluorescent tag. They found that control fibers without blood were negative. The blood-stained fibers were positive. So they concluded that human blood components are present in blood-stained fibers of the shroud. Now, an immunologist is going to look at this, and they're going to say, well, I think that you've demonstrated that blood components are present in the blood-stained fibers of the shroud, but I'm not so sure about the human part. I'm not really convinced that you've shown that this is truly human blood. And here's the reason they're going to say that. When you have these antibodies that are anti-human, that's how they're designated, that only refers to how the antibody was made. Anti-human just means you took a protein from a human, you put it into another species, and use that to make an antibody. It talks about how it's made, not what it reacts with. In immunology, there's something called cross-reactivity. So you can make it against a human protein, but in fact, that antibody may react with the same protein in a number of different species. And that's exactly what happened. So we now know some 40 years ago, or some 40 years in hindsight, the cross-reactivity is much greater than previously considered. These anti-human antibodies, they also react with primate blood, blood from rodents, from cat, dog, and a number of other species. So this was written up a few years ago. I'll just read this highlight here in the yellow. As discussed, cross-reactivity precludes a definitive assignment of human or even primate blood being present on the shroud. The blood is most correctly classified as species unknown. Thank you.
Thank you. I was curious on the Eucharistic miracle. One comment and one question. On the Eucharistic miracles, I applaud the rigor that you're suggesting. I also wanted to say that most of the miracles, in fact, the Vatican has taken really, really long to even state that they are miracles because it has gone through extensive testing, and it's only with the repeated patterns that they are seeing in this analysis that they are actually calling these miracles. So they've actually been super cautious about these things. So I wanted to ask you, when you recommend a kind of protocol and peer-reviewed science, don't you think that this is something that should stay within Vatican observatories and with these kind of protocols referred to them rather than keeping it in the hands of random labs?
Didn't quite understand that last part after you said kind of peer review.
Apologies. So I was saying Vatican is very, very conservative in calling these out miracles. So I do believe they've traditionally had very high standards for calling these out. Is there reason
so
for
I would
you to...
scientifically, I would, especially as an immunologist, I would say the rigor needs to be raised. Now, things like miracles for canonization of saints, there it's a very, very detailed, rigorous protocol. For the Eucharistic miracles, not so much. And if you look at what's been done, what's been published, there's only been 2 reports. The first one's back in 1970 for Miracle of Lanciano. There was another one recently in, I think it's not in a peer review, yeah, it's not. And some of the tests where they say it's blood, as an immunologist, I look at that and the control panels that I would like to see to feel confident about it are absent.
Shroud of Turin book that has recently been updated to compile everything that we make mistakes on. For example, it was the fire to which the shroud went through that actually burnt the fibers that suggested that the carbon dating predated.
Well, yeah, there's quite a different theories about the carbon-14 dating, why that would be off. The fire is honestly with this CO2 intake, that one's not too much in favor right now. Probably the one that's most in favor is that the area that they tested is not representative of the whole thing. So when they first were going to do the carbon-14 test, the protocol was we're going to take samples from 3 separate areas of the cloth. Okay, so that was what was agreed upon going in. When it was go time, what happened, they went to the bottom left-hand corner, they cut 1 sample, then cut that 1 piece into 3. Not the same as going for 3 separate sites. So they analyzed that section later and found it was different from the rest of the shroud. It had cotton interwoven into it, which the rest of the shroud is linen. It also contained dye, which the rest of the shroud does not. So a theory is that section was on the corner. They used to hold the shroud up over balconies at weddings and put pulleys on it to display it, all kinds of stuff. They think that corner was very easily frayed at some point, and they might have done a cut and repair dye job, and then it's distinct. But the evidence, it looks like it's different from the rest of the shroud. So that's one kind of push to redo the dating.
Actually, the doctors put out video with Rotonet that should suggest that what they said first is true. It's going to boost the mammoth shroud.
So, maybe I missed something, but so you were talking about. So, with regards to the AB,
Right.
So in the Eucharistic miracle, so you show the fungus is potentially on there, but so is there any reason to believe any of these miracles actually have the AB blood?
It depends on who you ask, but I would just say that you can get a positive AB signal without blood even being present at all. Now, with fresh blood, that's usually not an issue. But when you have artifacts that are contaminated, many common bacteria have it on there. So you can get that AB signal immunologically without blood even being present.
So the big picture takeaway, all of this should be taken with a grain of salt. Big grain of salt is what I'm hearing.
Yes.
Okay.
Yes, I would say that's very well put. Yes,
Okay.
a very big grain of salt, yes.
Okay.
Yes. Especially when you're trying to connect one relic to another and, relics and miracles, you have to have markers that are unique to do it. And when you know that AB contamination, AB is there on the bacteria, you can see this with contamination. You know these articles have been described as being contaminated. And also the miracles, it's pretty prevalent. So, yes, very big grain of salt. Yes, sir.
Thank you.
Yeah, thank you.
In terms of maybe more rigorous testing down the road, besides the AB antigen,
I'm sorry, but could you just speak up just a little louder?
I'm sorry. Yeah, thank you. Okay, so in suggesting more rigorous testing as they may happen down the road, and I would hope they do, besides the AB antigen testing, is it possible to test for RH factor? In fact, I think there are two RH. There's a RH and an RH2. Can those be tested?
Is this in terms of the shroud or?
Well, the shroud, or I mean,
The shroud RH has never been tested. You might see on the internet where it'll say it's been tested for RH, but it never has been done.
but it could be.
Could be.
All right, and the
be.
same then for the host Eucharistic miracles.
Yeah, sure. You could test those. And a lot of the stuff, when I talked about the antibodies, now we know 40 years later they cross-react, et cetera. Now there's tools to do that are bullet shots. I mean, human specific. Now, it won't even react with primates, so it's very doable these days.
Thank you so much for your talk. I found it
Just a
fascinating.
little. I'm sorry. I'm very hard of hearing. I'm sorry.
I said, thank you so much for your talk. I found it very fascinating. A few years ago, I became involved in a investigation of a purported Eucharistic miracle. And one of the tests we did that was, to me, very definitive was we simply took unconsecrated hosts, touched them to the blood, and then cultured them. And lo and behold, blood appeared on, it multiplied, as you would anticipate from a fungus or a bacteria, but typically speaking, human blood doesn't multiply itself across different wafers.
So sorry, but I missed that last part. I'm very sorry.
Typically, human blood does not replicate itself.
Oh, I see. I got
Yes.
you. I have you. I've got you. Oh, sorry. Yes, sir. Well, thank you.
Very pleased with the scientific. Oh, I'm very pleased with. I should eat this thing. No, I'll get better. Yeah? I'm getting hungry anyway. Microphone for dinner.
It's hard to talk into them. It is.
Very pleased with your scientific approach. My high-level question is: if we were ever able to explain these miracles scientifically, would it increase or decrease our faith?
Fantastic. It's a fantastic question. I think it would probably depend on the individual. I think
Right.
some, their faith is very solid, and they think the Eucharist at every Mass is a miracle.
Yeah.
And others, this would, I think, have a huge impact.
Yeah. So
And I think the church, especially when you want to, there are some questions, of course, faith is appropriate for, and some that science is appropriate for. But I think if you're going to approach it from the scientific angle, the science needs to be as absolutely solid as it
That's
can be.
right.
It needs to be airtight.
Yeah. Okay. Thank you.
Thank you.
All right. Thanks.
So this is something I'm not a theologian, but this is something a theologian would probably tell you or tell people, that there is a kind of misconception out there that what happens in transubstantiation, what happens at the Mass, is this actual physically turning into blood with hemoglobin and so on and so forth. and it's just somehow hidden from our sight, or we're not able. But that is, I think, not the Catholic position. The Eucharistic miracles aren't what is really happening ordinarily at Mass. And in a way, Eucharistic miracles are, in a sense, misleading people theologically as to what really happens at normally at a Mass. So I just
Yes, I think it's a very good point. Especially, this proves the real presence and things like that. I think that's an excellent point. Yes.
I wanted to ask about other Eucharistic miracles where they say this happens each year, and they have it in this glass case, Januarius or something.
I would have no explanation of that, like where it turns back to liquid, and there's no. Haven't really studied it that much, to be honest, but no explanation here for that. It's a good question.
Well, just a quick observation. Well, it's a transubstantiation, but the Eucharist is the body and blood, soul, and of Jesus under the full appearances of the physical bread and wine. So yes, what happens at Mass would really, this should not influence our belief in what happens at Mass at all.
I just want to make a quick comment about the blood of St. Januarius.
Just a little louder, I'm sorry.
I just want to make a quick comment about the blood of St. Januarius.
Yes.
It is possible to make a thixotropic mixture that when you shake it, it liquefies. Now, I'm not saying that that's what happens at the miracle of the blood of St. Januarius. I'm just saying that I can go into the lab and I can make something that looks solid, and then I shake it, and it goes liquid, right?
Hey, oh, okay. Great talk.
Thank you.
So I don't know much about Eucharistic miracles. I've listened to like one or two podcasts or something, but I think I remember somebody mentioning one Eucharistic miracle where there was heart tissue in the Eucharist. I don't know
Yes.
if you've heard of
Yes.
this, and what do you think
Yes.
about that?
Yes. So I had a slide when I had the HLA. It had it kind of off to the side. I didn't really talk about this that much in the talk because I have really no explanation as to how it could change into heart tissue. Now, others have questioned that. It's a question that naturally comes up if you look at this objectively, scientifically, as detective. Well, how much do we really know about those samples? Could it perhaps be any kind of substitution that might have occurred? Just a natural question that might come up. If you had these HLA markers in there, for example, because they show tissue sections of this, it's tailor-made to do the HLA. It would be an internal quality control check to ensure that no substitution had taken place because they should all, if genuine, should all be exactly the same. And that's the end game, to really show that. If you had that in place, it would become a moot point. It's an internal quality control check for that. As far as it actually being able to change into that, I have no scientific explanation for that. Yes, very good question. Thank you.
Thank you.
Thank you.